Protein synthesis is a two-step process. The two steps are transcription and translation. Transcription is where one gene is replicated in an RNA molecule. Translation is where an RNA molecule serves as a code for transformation of an amino acid chain. During the first parts of protein synthesis, an enzyme called DNA polymerase reads DNA and builds a strand of RNA based off of the code. Once this RNA is transcribed, the molecule then detaches the mRNA and leaves the nucleus and finds a ribosome to attach to. Once attached to this ribosome, the tRNA connects and reads the code of the RNA. This gene then translates to a polypeptide, aka the protein. DNA replication. DNA replication starts where helicase unwinds the DNA, breaking the hydrogen bonds. This creates a replication fork. There are two strands created, the lagging strand and the leading strand. The lagging strand is from 5 into 3, and leading is from 3 to 5. So they replicate the two processes. During leading process, it replicates with a primer made by primase binding to the 3 end of the strand. Polymerase cracks the new strand and binds to the strand at the side of the primer and adds a new base pairs. The polymerase adds Okazaki fragments between the primers. Exonuclease removes all RNA primers from the original strand and makes sure that there are no errors. Ligase joins the Okazaki fragments and forms a strand. Last is termination, which is where the polymerase gets to the end of the strands. The DNA then coils into a double helix, which produces two DNA molecules that contain an old strand and a new strand. How does salt and soap extract DNA from your cells? Soap and salt are added to a solution when extracting DNA because these ingredients are capable of breaking open both cell membranes, the one on the outside of the cell and the one on the inside of the cell that protect the nucleus with the DNA. And it lets DNA escape the cell.
What? No, pour that out.
Hey.
It says strawberry and bag with soap.
Strawberry in a bag and soap and soap.
No, I think it's the big. Wait, how do you know when the thing is
How does ethanol extract DNA from your cells? Ethanol is added after the soap and the salt because ethanol brings the DNA that has escaped the cells and into the solution to the top of the solution to be easily extracted and observed.
Say it twice, but you can walk in a little bit more. So you're gonna let this, yeah, you will. But let me just, Megan, let me just show you first. You're gonna let this sit for about 2 minutes.
Number 4. Restriction enzyme. Also known as enzyme nucleus. This little guy here is a restriction enzyme. Also, an enzyme nucleus. What I'm drawing here is a strip of DNA. These things here are hydrogen bonds. Again, we got hydrogen bonds here. And now we got codons. A, T, H, C, T, T. And then it'll go opposite on the other side. A, T, T. A, and the opposite would be T, 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 A, T, C, G, A. And now we are cutting this up like a restriction enzyme would do. Now this would be called a gene. Not those guys, but yeah. These things right here are phosphorylated bonds. Phosphorylated bonds. And this here is the gene. And then we got 40 base pairs. And the job of the restriction enzyme is that it cuts DNA in a specific sequence. Person 1. Person 1 gene has 40 base pairs, 40 BP, which is